Hello students, welcome back to Ligase Academy, your educational catalyst. Today we will be discussing about the last session in eukaryotic replication, which deals with nucleosome assembly during replication and also different models of replication. Coming to nucleosome assembly during replication, as we all know, a nucleosome consists of a histone octamer wound around with negatively supercoiled DNA. And one nucleosome consists of 145 base pairs. That is, 145 base pairs are found in one nucleosome. And a histone octamer consists of a tetramer of H3H4 associated with a dimer of H2A H2B, a heterodimer. And many chromatin chaperones are involved in the assembly and disassembly of nucleosomes at replication fork. And one of the chaperone is FACT, that is, facilitates chromatin transcription. And this chaperone will disassemble H2A, H2B in front of replication fork. And the remaining H3 and H4 are disassembled by MCM complex, that is helicase. And this fact has another function. It also reassemble H2A, H2B behind the replication fork. But the assembly of H3 and H4, it requires another chaperone, that is CAF1, that is chromatin assembly factor 1. So there are some peculiarities for CAF1. This CAF1 translocates newly synthesized H3H4 in the cytosol to the nucleus and assemble it on daughter DNA randomly. So even though replication is semi-conservative, inheritance of histone is dispersive. That is, CAF1 only recognizes newly synthesized H3H4 because these specifically bind acetylated lysines in the H3 and H4. So the enzyme required is histone acetyl transferase, which is only localized in the cytosol. This is the reason. Mitochondrial DNA replication, it is based on strand displacement model of replication. That is, only one strand is replicated at a time. So, there are two origins present here, OH and OL. That is, OH is the origin for heavy strand synthesis. That is, in OH, that the strand contains guanine residues content that increases the density, and OL is the origin for light strand with poor G residue. So the replication begins at OH, and when replication is 2/3 completed, the oil is exposed and a replication of light strand begins at opposite direction. That is the thing happening in strand displacement model. And there is no dedicated primase present here. The mitochondrial RNA polymerase function as primer and they produce primary transcript, and single strand maintenance protein is mitochondrial SSB protein, and a dedicated 5' 3' helicase that is called as twinkle is present here, and a dedicated DNA polymerase is pol delta, and the overall result after the strand displacement model is the formation of hemicatenins, and they are resolved by topoisomerase 1. So this is the schematic representation of strand displacement model of replication, and in the image it's clear that from the OH replication proceeds and one parental strand will displace out to form D loop that constantly expands. And when the replication is 2/3 completed from the OH, the oil is exposed and the replication of light strand begins at opposite direction. So after the two replications, the result is the formation of hemicatenins, that is circular DNA connected to each other by a small single strand regions. So they are resolved by topoisomerase 1. So we will get two daughter DNA. So the replication in mitochondria by strand displacement model as unidirectional, asynchronous, asymmetric, and continuous. And also there is no Okazaki fragment formation present here. So that's all about Strand displacement model of replication. Rolling circle model replication, and this type of replication is observed in plasmids and bacteriophages, and similar to Strand displacement model, this model also contains two origins, that is double stranded origin DSO and single strand origin SSO. And the initiator protein, the present here is RUP-A endonuclease. That is, they belongs to a family of HUH endonuclease. That is, they contain HUH motif, that is two histidine residues separated by a hydrophobic residues. So they recognizes the origin, and their catalytic motif contains a critical tyrosine residue that carries out trans-esterification reaction. That reaction is similar to topoisomerase type 1. So this is the reaction occurring in origin when initiator protein binds. So origins are recognized by their three-dimensional shapes. Mainly double strand origin contain a cruciform DNA. That is, they are formed by a palindromic sequence in a double-stranded DNA. But when in the case of single strand origin, they have a higher pin formed by palindromic sequence in a single strand DNA. So first the replication begins at double strand origin, and the double strand origin contain two sites, bind and nick. And bind is the region where RUP protein, that is initiator protein, is recruited, and nick is the region where RUP protein nicks the DNA. So let's discuss the steps occurring in the rolling circle model of replication. So the RUP initiator protein will recruit it into the bind region and it nicks the double stranded origin. Then they physically attaches to 5' end of nicked strand, and the free 3'OH is the primer and is extended by the host DNA polymerase 3. And as replication from double strand origin continues, the nicked strand is looped out, and a second esterification by RUP on double strand origin will release a daughter duplex and the replication intermediate called replicative form. So the replicative form is covalently closed circular single strand DNA. So next, the single strand origin is exposed on replicative form, and primary transcript from RNA polymerase host function as primer for replication of replicative form. So thus two second daughter duplex thus forms. So this rolling circle model is unidirectional, asynchronous, asymmetric, and continuous. Like single strand displacement model, here also there is no formation of Okazaki fragments. So that's all about rolling circle model of replication. The RUP protein will binds on DSO, and they physically attaches to the 5' end, and the free 3' OH extended by DNA polymerase 3. And as this replication from DSO continues, the nicked strand is looped out and a second esterification reaction occurs, and intermediate called replicative form. So after that replication occurs for this replicative form in the same manner, and thus we got two daughter duplex form. That's occurring here. Okay. And the replication of chloroplast genome is same as double strand displacement model. Here also there will be two displacement loops are formed. That is the difference. Okay. That's all about replication. So a broad topic ends here. Hope you all understood about replication in detail. If you have any doubts regarding this section, please let us know through the comment section. And if you find this video is useful, please don't forget to like and share to your friends. And for more related topics, please subscribe to our YouTube channel like Academy, your educational catalyst. Thank you.